Welcome to the video for Lecture 1.2, in which we will be covering the scientific method. This scientific method meme was brought to you by one of my previous students. Our learning objectives for this video are to learn and apply the main steps of the scientific method to develop a scientific hypothesis. We will also learn about what the different variables in scientific experiments are and how to identify them. Before we go into the scientific method, let's talk about how we perform scientific inquiry, or how we think about science. Science involves logical thinking. There are two methods of arriving at logical conclusions: inductive and deductive reasoning. Inductive reasoning is a narrow to broad approach. It begins with specific observations. From these observations, we find a pattern. We develop a hypothesis about the pattern, and then we draw a general conclusion from our tests of the hypothesis. For example, I can observe that eating chocolate makes me happy. I see a pattern in which my friends are also happy when they eat chocolate. From there, I develop the hypothesis that if I give chocolate to someone, they will be happy. I can test this, and if people are happy when I give them chocolate, I can draw the general conclusion that chocolate makes people happy. Deductive reasoning, on the other hand, has a broad to narrow approach. It begins with a general rule, and we develop a hypothesis based on that rule. We test that hypothesis, and we can then make a specific conclusion based on the results. For example, a general rule is that cats like to eat fish. I can make the hypothesis that since tuna is a fish, cats like to eat tuna. I can test this by giving tuna to cats and seeing whether or not they like eating it. I can then draw the more specific conclusion that cats like to eat tuna. Now let's talk about the scientific method. The scientific method begins with an observation. Observations can come in many forms. For example, it can be something you personally observed or an observation you made from reading. After making an observation, a question is then formed based on the observation. The hypothesis is the suggested explanation that addresses the question. You may have also heard the hypothesis being defined as an educated guess. An important aspect of the hypothesis is that it must be able to be tested, and therefore must be capable of being disproved. Based on the hypothesis, we can make a prediction on what would occur if the hypothesis is tested. We then test the hypothesis with an experiment. If the results of the experiment disprove the hypothesis, then we reject it. If the results do not disprove the hypothesis is not rejected. As we learn more about biology and make new observations, we will generally form new hypotheses or revise old ones. For example, the observations we make from our results will lead us to form new hypotheses. The way we design our experiments to test the hypothesis is very important. All experiments will have at least one independent and dependent variable, and multiple control variables. The independent variable is generally the variable that is being tested and manipulated. It is called the independent variable because if there are multiple independent variables, manipulating one will not affect the other. The dependent variable is generally the variable that is being observed or quantified. It is called the dependent variable because it can change depending on how the independent variable is manipulated. The control variable would be any variable that is not being tested by the experiment, and therefore is always constant. All experiments must also have at least two groups. One group is the experimental group, which is the one that is being tested for some independent variable of interest. Generally, this means that this is the group that is receiving some type of treatment. The other group is the control group, which essentially just provides a standard that the experimental group is compared to. Now let's do an example. Say I planted some tomatoes and they seem to be growing well, but after a few days of 100-degree Fahrenheit weather, they are not growing as well as before. I can then ask the question, why are my tomatoes not growing well anymore? One hypothesis I can make is that tomatoes do not grow well when the temperature is too hot, specifically at temperatures over 100 degrees Fahrenheit. I can then make the prediction that if I grow tomatoes in a room at 100 degrees Fahrenheit, the tomatoes will not grow as well as tomatoes grown at a more moderate temperature that I define as 75 degrees Fahrenheit. In this experiment, I want to look at the effect of temperature on tomato plant growth. Since I want to know what the effect of temperature is, that is the variable I am manipulating, and so my independent variable is the temperature. I am observing the growth of the tomato plant by measuring the height of the plant, and so my dependent variable is the height of the tomato plant. I would then keep all the other variables constant, such as the amount of water they receive, when they are watered, the soil they are in, and the amount of light they get. There are even more control variables that I haven't listed. For the sake of simplicity, I will just have two groups. My control group would be the tomatoes grown at 75 degrees Fahrenheit. My experimental group would be the tomatoes grown at 100 degrees Fahrenheit. When the experiment is finished, I will compare the growth of the two groups by measuring the height of all the tomato plants. If the height is the same, or the tomatoes grown at 100 degrees Fahrenheit are taller, then my hypothesis is disproven. However, even if the tomato plants grown at 75 degrees Fahrenheit are taller, that does not necessarily prove my hypothesis. It would demonstrate that in general, tomatoes grow better at 75 degrees Fahrenheit than at 100 degrees Fahrenheit, but there are still many other possible explanations for why my tomato plants in particular did not grow well. For example, perhaps I forgot to water them at some point, or I did not water them enough because I did not take the heat into account. That's it for Lecture 1. In this video, we went over inductive and deductive reasoning, and how both are ways of logical thinking that are used in science. We also discussed the scientific method, which always begins with an observation. The observation leads to a question, and our hypothesis is an educated guess for what the answer to that question might be. A prediction is then made based on the hypothesis, and we can then design an experiment to test the hypothesis. All experiments involve independent, dependent, and control variables, and there is always at least one control and experimental group. In the next video, we will begin Lecture 2 and start learning about the molecules that are important for life.